Alright, so let us continue with the polysaccharides. So, these are some of the functions and descriptions of polysaccharide sugar. So, some of them are known as storage polysaccharides. So, when you speak of storage polysaccharides, so they store carbohydrates. So, they are also energy storage in the case of starch and glycogen. And of course, there are also structural polysaccharides. So they used to provide protective walls or lubricating coat or lubricative coating to cells, like for example, cellulose and mucopolysaccharides. And there are also what we call as structural peptidoglycans, like for example, bacterial cell walls. Now, for the nomenclature of it, we have homopolysaccharide. Homopolysaccharide are polysaccharides that are made up of one type of monosaccharide unit. So when we speak up of one type of monosaccharide unit, for example, all of them are glucose or all of them are galactose or all of the monosaccharide composing the polysaccharide are, let's say, fructose, for example. So that's why the term is homo. So there should only be one type of a monosaccharide unit. Now, for the heteropolysaccharide, a polysaccharide that is made up of more than one type of monosaccharide unit. So, for example, there is glucose, and then there is fructose, and of course they are all connected via the glycosidic bond. So again, it is a homopolysaccharide and a heteropolysaccharide. So let us now look into the first example, starch. No, so starch is a polymer consisting of the glucose units. So it means that if it is consist only of the glucose unit, then therefore it is polysaccharide. So starches and other glucose polymers are usually insoluble in water because of the high molecular weight, but they can form thick colloidal suspensions with water. So starch are insoluble in water, but they do combine with it, forming a colloidal suspension. So starch is a storage compound in plants and made up of glucose units. So as I mentioned, it is a homopolysaccharide that is made up of two components, amylose and amylopectin. So meaning that the starch is composed of amylose and amylopectin. So most starches 10 to 30% amylose and 70 to 90% amylopectin. And amylose is a straight chain structure formed by 1,4 glycosidic bonds between the alpha-D glucose molecule. So let us look into the example. So what makes it 1,4? It is 1,4 because the first carbon of the glucose is attached to the fourth carbon of the next glucose. So that's why the term is alpha 1,4 glycosidic linkage. The example on top of the illustration is an amylose. And then on the other hand, the amylopectin is consist of the 1,4 and 1,6 glycosidic linkage. What makes it 1,4? So let me draw it again so that you'll be guided properly. So this is 1,4, that one. Okay. So this is also 1,4, 1,4. So the one that I explained a while ago is also 1,4. Okay. So it's the carbon. It's the carbon that is attached to the oxygen for the glycosidic bonding. Now, in this case, there is 1,6. So 1,6 is when there is a branch. When there's branching in amylopectin, so they can only do that on the sixth carbon. And Because of that, the first carbon of the glucose is attached on the sixth carbon, yung plug pole, no? Plug pole carbon in this case, in this case, no? So later on, I'll be discussing on how to create this kind of formula. So this is 1 and this is 6. So that's why it is known as 1,6 alpha 1,6 glycosidic linkage at branch point. All right. So let us now move on to the next. So the amylose chain forms a helix. No, so a helix is like the DNA helix. So this causes the blue color change on a reaction with iodine. So one of the tests that you'll be doing for the laboratory for test for carbohydrates is the utilization of iodine. No, so um, if there's a reaction to it, then we can say that there is a presence of carbohydrates. So amylose is poorly soluble in water but forms micellar suspension. Micellar suspensions are found at the bottom of the container. And then amylopectin, a glucose polymer with mainly alpha 1,4 linkages, and of course, branching out as alpha 1,6 linkages, the one that I have shown a while ago. Then the amylopectin causes a red-violet color change on reaction with iodine. So the other one, the amylose, can react to iodine forming blue color. Now, on the other hand, if it is amylopectin that is present on the sample, it is not blue, but it is red-violet color. So this change is usually masked by the much darker reaction of amylose to iodine. So another example of polysaccharide is glycogen. So I think as I have mentioned this before to you, no, it is a storage polysaccharide in animal. So yung starch a while ago is a storage in plants. This one is a storage polysaccharide in animals. So glycogen constitutes up to 10% of liver mass and 1 to 2% of muscle mass. So that's how big the contribution of the glycogen in the liver as well as in the body. So glycogen is a stored energy for the organism. So it is a store energy because once we accumulated lots of glucose from the food that we eat, so it will be regulated by the hormone insulin, and then it will give signal to the organ to absorb the glucose. And as they do that, as the liver do that, it is converted to polysaccharide for storage in a form of glycogen. So similar in structure to amylopectin, only difference from starch is the number of branches. So alpha 1,6 branches every 8 to 12 residue. And like amylopectin, glycogen gives a red-violet color with iodine. So let's say, for example, you are going to test amylopectin in glycogen. So you'll be able to have the same result just like in the starch in amylopectin, which is red-violet in color. So this is the structure of a glycogen molecule. So same thing, no? It is a 1,4 glycosidic bond and 1,6. There you go. Glycosidic bond in the branch. So another is cellulose. I am sure that you are very much familiar with this because this is a component of the cell. So the beta glucose molecules are joined by condensation. So when we speak of condensation, it is the removal of water. So once we remove water from the glucose molecules, so they will tend to form polysaccharide or disaccharide and then disaccharide to polysaccharide. So the removal of water forming 1,4 glycosidic linkages. So the glucose units are linked into straight chains, each around 100 to 1,000 units long. So when we speak of 100 to 1,000 units long, that is 100 monosaccharide sugar linked together by the glycosidic linkage up to 1,000 units. So weak hydrogen bonds form between parallel chains, binding them into cellulose microfibrils. So these cellulose microfibrils arrange themselves into thicker bundles called microfibrils. So the cellulose fibers are often glued together by other compounds such as the hemicelluloses and calcium pectate to form a complex structure such as plant cell walls. So because of such combination, no, it leads to the formation of the plant cell wall. The plant cell walls are very thick and rigid because of the presence of the cellulose. Because of the beta linkages, cellulose has a different overall shape from amylose, forming extended straight chains which hydrogen bond to each other, resulting in a very rigid structure. So this is the explanation what makes cell wall rigid, no? So as we all know that part of the defense mechanism of plants, since they do not have any um, parts that literally move just like the animals, wherein animals can defend themselves. Now plants cannot do that, so instead they ensure that their cells are very rigid and very strong, that um, other animals will not uh, will not eat them that easily. So cellulose is an important structural polysaccharide and is the single most abundant organic compound on Earth. So it is the material in plant cell walls that provide strength and rigidity. So wood is 50% cellulose. So just imagine that those wood that we use in constructing houses, in making of furnitures, no? So that's 50% cellulose. So that's how hard the cellulose are. So most animals lack the enzymes needed to digest cellulose. So as I mentioned, I think I mentioned this before, that since we cannot digest cellulose, no, so it serves as fiber in our body that clean up our digestive tract. So although it does provide needed gauge, no, or dietary fiber to stimulate contraction of the intestines and thus help pass food along through the digestive system. So some animals such as cows, sheep, and horses can process cellulose through the use of colonies of bacteria in the digestive system, which are capable of breaking cellulose down to glucose. So that's the advantage of grass-eating animals, just like cows, sheep, and horses, wherein they have this kind of bacteria in their body that help them break down uh, the cellulose into glucose. So since it will be broken down into glucose, so it can serve now as the energy of that particular animals. So ruminants use a series of stomachs to allow cellulose a longer time to digest. So since it is rigid, it is a rigid structure. So it is, even if they have bacteria, it will not easily be digested. So instead, they have this series of stomachs, no, so that it will take a longer time to be digested. So some other animals such as rabbits reprocess digested food to allow more time for the breakdown of cellulose to occur. So when we speak of reprocess, uh, so they have to undergo the process of digestion again. So cellulose is also important industrially from its presence in wood, paper, cotton, cellophane, rayon, linen, nitrocellulose or gun cotton, photographic films like cellulose acetate. So there are a lot of economic importance of cellulose from plants. Okay, so this is the structure of a cellulose. So there's no branching out, no, but it is one for glycosidic linkage. So another one is chitin. So chitin is a polymer that can be found in anything from the shells of the beetles to webs of spiders. It is present all around us in plant and animal creatures. So it is sometimes considered to be a spin-off of cellulose because the two are very molecularly similar. Closely similar, but they are not exactly alike. Cellulose contains a hydroxy group and chitin contains acetamide. So chitin is unusual because it is a natural polymer or a combination of elements that exist naturally on Earth. So usually polymers are man-made. However, there is this natural polymer. So crabs, beetles, worms, and mushrooms contain large amount of chitin. So chitin is a very firm material and it helps protect an insect against harm and pressure. Actually chitin are found among the exoskeleton of insects, no? So that's why even if we stamp on the insects, they don't usually or they don't easily die because of the presence of the chitin. And there are also other people who eat insects, no? And some of them are saying that it tastes like sugar or it's a bit sweet and so on. So simply because it is a bit sweet because of the presence of chitin, no? And this chitin is a polysaccharide sugar. So when you digest it, if you have the ability to do so, no? So you can be able to taste uh, that sweet taste. So this is how it looks like. So you have there the sugar at the middle, no, at the center, and then there is the acetyl group. So the acetyl group, the NH, C double bond O, and CH3. So another one is inulin. So inulin is stored in tubers of dahlia and artichoke and in the roots of dandelion. It is also found in onion and garlic. So inulin has a molecular weight of about 5,000 and consists of about 30 to 35 fructose units per molecule. So that's dandelion. Sometimes dandelion, but dandelion. All right. So it is formed in plants by eliminating a molecule of water from the glycosidic OH group on carbon atom 2 of 1 beta D glucose unit and the alcoholic OH group on carbon 1 atom 1 of the adjacent beta D fructose unit. So for you to be able to picture this out, it looks like this. So you have there the arrangement or the way the fructose are interconnected by the glycosidic bond. And then we also have pectin. So pectins are found as intercellular substances in the tissues of young plants and are especially abundant in ripe fruits such as guava, apples, and pears. So that's why the fruits are becoming ripe, no? Nahihinog yung mga fruits. It is because of the presence of pectin. So at first, it is in a very small amount, intercellular substance, no? However, as time goes by and the the fruits grow, so there are more pectins. And then at the end of it, if it is already ripe, it means that there are a lot of pectins already. So pectin is a polysaccharide of a D-galacturonic acid where some of the precarboxyl group are either partly or completely esterified with methyl alcohol and others are combined with calcium or magnesium ions. Chemically, they are called polygalacturonides. All right. So it looks like this. So still it is glucose. However, there is this COOH or carboxylic acid. And we also have COOCH3. Unlike in other structure na there are no other attachments in glucose but the usual attachment. Okay, so that ends the last classification of carbohydrates. So this time let us talk about Haworth projection. Okay, so as useful as the Fisher projection is, if you will still recall on the think on the first uploaded video, so there's a discussion about Fisher projection. It gives a poor sense of the real structure of carbohydrates. The Haworth projection is a way around this limitation that does not require you to try to convey the complete 3D image of the molecule. So, sugars in Haworth projection can be classified according to the ring size. So, Haworth projection looks like cycloalkane, the one that gives shape, no? So, in this case, you have the, if it is composed of 5 carbon, so you call it as puranosis. And if it is composed of 6 carbon, it is known as pyranosis or pyranose and puranose. And it looks like this, no? So pyranose on the left side of the slide and puranose at the right side of the slide. So you have here pyran and puran. So pyran and puran, they're pyranose and puranose, no? For the alpha D glucose, you have there, yeah, it's an alpha D. And then the other one, in alpha D glucose, there are 6 carbon in glucose. So that's why there are also 1, 2, 3, 4, 5, 6, 6 sides, no? But there's the flagship carbon. So later on, I'll be discussing that to you. Then for the alpha D fructose, so it is puranose, only composed of 5 carbon atom. And then you also have there another example of puranose, alpha D ribose. So puranosis, we divide Haworth projections into two classes, puranosis and pyranosis. As I mentioned, the puranosis or pi-member ring, hemiacetals are drawn with oxygen at the top of the pentagon. So when you draw it like a pentagon, but the oxygen is found on top of it. So the horizontal band of at the bottom is assumed to be coming out of the plane toward you. Thus, the pi-member ring is in a plane perpendicular to the page. So later on, I'll be showing you the illustration. So vertical lines are drawn on each carbon to indicate attachments above and below the plane of the pi-member ring. So in solution, fructose ribose and deoxyribose will exist as pi-member puranose rings. The puranose rings resemble the cyclic either called puran, and a puranose form the sugar ribose is a good example. So um, all you have to do, all you have to remember here is that puranoses are composed of pi-member ring. Okay? And then you have to remember that the oxygen is put on top of your illustration. Okay, so how about pyranose? So pyranose 6-member rings have a slightly different but quite similar Haworth projection. So a hexagon is placed so that one horizontal band runs along the bottom. The oxygen is in the ring is placed at the upper right, at the extreme right. In solution, glucose, galactose, and mannose will exist as 6-member pyranose ring. So let us now look into that. Okay. So what are the rules? Draw either a pyranose or puranose ring depending on the sugar. So if glucose will be converted into a Haworth, then there should be 6 carbon. If for example you'll be doing fructose, no? So all right. So let us now look into this. So if it is Fisher, OH to the right. If it is Haworth, if it is found at the right, yung OH, then it is found at the bottom of the ring. If it is OH to the left, OH must be above the ring. Now for anomer, anomer um, is the Haworth projection, no? The one with the shape. For anomer, place the anomeric carbon OH opposite the flagpole carbon. So anomer is the first carbon, anomeric carbon, no? So you have to look into where you are going to put the OH, whether it is found on top or at the bottom. However, there is a sign there. So if it is placed on the same side as the flagpole carbon, the flagpole carbon is located on top like this. So If the anomeric carbon OH is same side as the flagpole, nakataas na ganito, we call it as beta anomer. However, if it is the OH is down, so you call it as alpha anomer. So anomeric carbon, the new chiral center created when the sugar ring is formed, and anomers, the two new sugar stirring isomers created by ring closure. So this is how you convert Fisher projection to Haworth projection. Now, in Haworth, you can make two. Either you do the alpha version or the beta version. So let us now look into this. So this, you have 6 carbon. So you'll be making the shape. So the first carbon or the anomeric carbon, this is the anomeric carbon. Okay, so see there, then 2, 3, 4, 5, and 6. And then after that, the OH group on the C5 adds across the C double bond O. So it went there. See the arrow. Okay. And then this one, the sixth carbon here, will become the flagship. No? Will become the flag. No? So it is now looks like a flag like that. Okay. And then after that is the oxygen will be bonded with the first carbon. So because of that, this H will be mixed or combined chemically with this O. So since it will going to receive H from the OH, so there will no longer be double bond. So it will be removed. It will just become single bond because oxygen can only take this, no, this one. Okay, so at the end of it, so there are two possible stereoisomers. First is alpha D-glucose, okay, and the other one is beta D-glucose. So this one is an opposite, one is down, and this is up. This one is up and up. All right. So let us look into this. I think this is easier. Converting the Fisher projection to a Haworth projection. So D-glucose again. So if it is right side, then it is down. If it is left side, then it is up. So let's look into that. So you have here, it is alpha peranose. So 2 is left side, left. So if it is left, it is, no, it's right. Okay, if it is right, this is right side, then it is down. This one down. This is left side is up. This one. And then this is again right, then this is down, and this is right, and fifth is here. Okay. So there's no more OH here on the fifth because it underwent a reaction here in the oxygen. So ito yun, no? Punta na dito. Alright, so if OH is on the right, it is a D sugar, CH2OH up. So all D sugar, it is found here on top. Pero if it is L, this one is found here at the bottom. Okay. Now for the alpha anomer, it has OH for C1. So it's down. If it is beta, OH is up dito. Right? So, yan. Okay, so that ends our discussion on carbohydrates. So, I hope that you're going to watch this video so that you'll be able to answer all your activities. Again, the submission is on the schedule for the week, and it should be submitted in hard copy. So thank you so much.